Welcome to Ask the Expert. This is a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event and we'll post it on the Sugar Science YouTube channel shortly after the presentation. If you have any questions for our guests, please feel free to put them in the chat or raise your hand at the end of the presentation. And today we have as our guest Dr. Ron Weiss, coming to us from MIT. And Professor Weiss is one of the pioneers of synthetic biology. He's been engaged in synthetic biology research since 1996 when he was a graduate student at MIT, and where he helped set up a wet lab in the electrical engineering and computer science department. After completing his PhD, he joined the faculty at Princeton and recently returned to MIT to take on a tenured faculty position in the Department of Biological Engineering and Department of Electrical Engineering and Computer Science. So the Weiss Lab seeks to create integrated biological systems capable of autonomously performing these useful tasks and to really elucidate the design principles underlying complex phenotypes. So sort of in a nutshell, cells sense their environment, they process information, and they continuously react to both internal and external stimuli. One cell that does that, that's near and dear to our hearts, is the beta cell. And of course, it's alpha cell, it's neighbor in the pancreas. So it'll be interesting to talk a little bit about how some of these concepts might extend that realm. And then the construction of synthetic gene networks really can help improve our understanding of such naturally existing regulatory functions within cells like those I just talked about. And they also sort of enable a wide range of new program cell applications. So the Weiss Lab uses computer engineering principles of abstraction, composition, and interface specifications to program cells with sensors and actuators precisely controlled by analog and digital logic circuitry. That's a huge mouthful, but we're going to talk about Dr. Weiss's work and his exciting new paper that was just published in Science in July 2020, and it was titled An Engineered Protein Phosphorylation Toggle Network with Implications for Endogenous Network Discovery today. So thank you so much for joining us. We're looking forward to hearing about your work and what's going on in your laboratory.
Thank you for the invitation. Pleasure to be here.
Great. So did you want to talk a little bit about what's going on over in the laboratory?
Yeah, and so I have a few slides prepared. I can talk, without slides, or I can present a few slides. It's up to you.
Slides would be great.
Okay, wonderful. So I'm going to share the slides. Let's see. So what I wanted to talk today about is actually two projects that are related to diabetes and pancreas, and one of them has to do, one vignette will have to do with the manuscript that was just recently published, but I actually want to start by talking about something that's been kind of near and dear to me essentially for a quarter century now, which is once I got into this field of synthetic biology 25 years ago as a graduate student, I kind of realized that one thing I would really want to do is be able to program cells to create tissues by design. And so actually going back 25 years ago as a graduate student, I was looking at ways by which we can engineer cells to create spatial patterns and then use these spatial patterns as a means to guide cells to differentiate into particular tissues. And started out working by creating patterns in bacterial systems by getting bacterial cells to send messages, chemical messages back and forth. But around now about 15 years ago, we began to shift away from bacterial systems and really focus our attentions on mammalian cells. And the question was, how can we program stem cells to create these tissues by design? And now these are essentially called organoids. There's been a lot of really exciting developments in the field of organoids. So one of the things in particular that we're interested in the lab is understanding how to create two kinds of organoids. I'm going to talk about a liver organoid, but also with the intent of creating pancreatic organoids. And you can imagine that if we can create pancreatic organoids out of the patient stem cells or the patient's induced pluripotent stem cells, we might be able to actually engraft those, transplant that into the patient, and then engraft them as a way to help address some of the issues with diabetes. And so you can imagine creating these kinds of organoids, I mean, is rather involved. And there's a lot of inherent biological complexity in these types of tissues and organoids, including the pancreas, where there's complex pathways that control the three-dimensional architectures of the pancreas, the complex pathways that control how pancreatic development actually takes place, and there's complex pathways that are involved in controlling pancreatic metabolism. And one of the kind of foundational things that we are working on is this notion that synthetic biology actually provides the appropriate level of sophistication that would allow us to really gain control over the development of organoids such as the pancreas, right? So, and this relates both to the work that we're doing, directly on organoids, but also to the sense and respond mechanisms that we discussed in the recent manuscript. And so, a lot of what we do in synthetic biology is to create circuits that can sense what's going on either inside the cell or outside the cell, and then process that information, make decisions about what is being sensed, what's going on, what's the internal state of the cell, what's going on outside the cell, and then regulate what happens inside the cell. So what's the therapeutic function that we might imagine doing inside the cell, or perhaps secrete something outside the cell in response to changing conditions. So you can imagine situations where you have abnormal blood sugar levels, right, that are detected. And so the question might be, can we engineer cells to detect that and then be able to respond by secreting, insulin, for example, right? But what I want to talk about, and so that relates to kind of the mechanisms that we're putting together, the fast sense and response mechanism that we're putting together that we published on. But actually what I want to briefly talk about first is, can we use these sense and response mechanisms to control the development of organoids such as the pancreas? And so, for example, can we start with these induced pluripotent stem cells, program them to proliferate, create patterns, and then ultimately create a desired type of organ using synthetic biology? And so what we've been able to do with folks from my lab and colleagues such as Linda Griffith is be able to take these induced pluripotent stem cells and genetically engineer them to actually create liver organoids. Okay, and I'm going to tell you in a second why that's relevant for pancreas. And so these liver organoids that we engineer start with iPSCs, and then they develop into endoderm and mesoderm. Let's focus on that. And then the endoderm becomes hepatic, so create the hepatocytes and other cell types that exist in a liver, as well as mesoderm that creates things like endothelial cells and vasculature. So we end up getting these vascularized liver organoids that look like this, and they become really rather large. And
Wow.
These are on the order, on the order of centimeters or so. And by the way, I'm happy to take questions now during these, or questions after. And so we create these vascularized liver organoids. They have all the cell types that are known to exist in liver, fetal liver, and they have the right, liver function to a large extent. And so the question is, can we take something like this, that we now can create a liver organoid, and can we ultimately create a pancreas? And so what does that mean? So we'll start with iPSCs, and this is now work in progress, where we want to take what we've been able to demonstrate with liver and demonstrate that for the creation of basically customized patient-specific pancreatic organoids. And so the mesoderm that develops in the vasculature will remain the same, but on the endoderm part, where it used to go to hepatic lineages, we will embed these circuits that guide the differentiation towards pancreatic lineages, such as getting endocrine cells, exocrine cells, beta cells, alpha cells, acinar cells, and so on. And so we've been working on a variety of sense and response mechanisms that would actually sense, for example, that the particular subpopulation of cells have become endoderm, and then drive them towards pancreatic lineages by expressing the appropriate cell fate regulators that actually take them down that path. And so that kind of leads us. So that's a huge focus of work in our lab, and that is a sense and response mechanism that works on the order of hours or so. And so in the recent publication, we've also been working on mechanisms that would work very quickly, so on the order of seconds. And so that would be relevant, for example, if we're able, if we're embedding sensing mechanisms that detect, there's aberrant glucose levels, and we want to be able to secrete insulin in engineered cells. So we need to have something that responds very quickly, not hours, but rather seconds. And so
Yeah,
for that,
it's even like, milliseconds, I think, right?
I think you can, if you do it with responses over the course of seconds, that's actually, I think, okay. With respect to, to these kind of glucose sensing insulin secretion. You can't do it, in tens of minutes, but I think if you have response in several seconds, I don't know, even possibly a minute or so, I think you probably would be okay.
Yeah,
even several minutes.
Yeah, because I mean, in the natural, situation, it's I think, the whole sequence, the secretion and detection of what's been secreted is, in milliseconds or seconds. Yeah, I mean, right now, what do people have? They have a CGM that basically takes like 5 minutes to really detect blood sugar. So yeah, it's a big improvement.
Yeah. Yeah, and so this would be done rather than an external device. This would be done as something that we genetically engineer cells in your body, and they would be the ones that are constantly monitoring, and and they can make very rapid decisions with the kinds of protein. The key here is that we're using protein-protein interactions.
Right.
And because we're not relying in the other projects, and most of the things that we do in our lab, and most of the things actually in synthetic biology, they require sensing, and then the response is based on the production of new proteins. And the problem is through processes called transcription and translation. And the problem is that the production of new proteins really is, it takes a long time.
Yeah.
You need, in order to be able to generate and make enough new proteins, that really is on the order of tens of minutes to hours.
Yeah.
To get them to produce enough proteins, whereas here, we're talking about protein-protein interactions.
Yeah, much faster. It's really, it sort of follows the term, the switch, right? It's really quick. Yeah.
And so the way that we've been able to demonstrate these protein interactions is that you'll get an input signal. And so this input signal could be, for example, your blood sugar level is too high. And so then there's a set of downstream interactions. So, what would happen is, what we've been able to demonstrate specifically is that with an input signal, and that's actually osmolarity from the outside, we're able to show that these proteins inside the cell get phosphorylated. So they change the phosphate groups on the proteins. And as a result of that, the upstream protein will then bind, it's phosphorylated, will bind the downstream protein. So that's due to this, external sensing of osmolarity, followed by phosphorylation of the upstream, followed by regulation of the downstream element. So we have this cascade of events that takes place, and we've been able to show that this mechanism here can be used to build all kinds of logic, like in the way that you have computer logic that works on and or not type of operations. We've been able to demonstrate that we can engineer biological pathways and biological interactions through protein-protein relationships and interactions that perform an OR operation. An OR operation would be either this input is high or this input is high, and that ends up resulting in phosphorylation of a downstream element. So we've been able to show that. We've been able to show things like NOT gate. So NOT gate means that if the upstream element is phosphorylated, then the downstream element should not be phosphorylated, and vice versa. If the upstream element is phosphorylated, then the downstream element should not be phosphorylated. So it's doing the logical NOT or inversion operation. And so the critical thing about these logic gates, the OR and the NOT logic gates, is in principle that allows you to build any kind of logic control circuit that you want. So if you want to have the ability to detect very quickly, these aberrant conditions, and maybe not just one, maybe there's, it's more complex than that. You can build logic operations that can detect kind of this multi-state variables. And we've been able to then show that we can take these basic control units, and I'm specifying that here as OR and NOT gates and other kinds of gates, and actually build them inside cells. And I'm not going to get into all the details of all of these interactions. This is a fairly complex network, but this is a network that we've been able to engineer in yeast that can sense sorbitol levels from the outside, or can sense another molecule called IP, and basically can set a switch. So this becomes like a 1-bit memory inside these cells. So they can remember, that we sense this level, glucose or something else, or hormones in your body. And that allows setting of the switch, and the switch is actually very fast. So the switch, here you can see the protein, fluorescent protein molecules going in and out of the nucleus of the cell. They go in and out of the nucleus, and this is shown reactions in minutes, but we've actually in the paper been able to demonstrate that we can sense an event that just 1 second, that took place just for 1 second, and we can have a permanent memory of that event, even though it was only like super transient.
This is fantastic.
And so that one.
This is really amazing.
And so, thank you. And so, so we're hoping now to be able to use that for engineering cells that we can then embed in your body that can do this very fast sense and response, with an eye, perhaps towards diabetes is certainly something that we've been thinking about. Also, other kinds of, misregulation, if you have certain cytokines or chemokines that are misregulated, or maybe there's like signs of an impending heart attack or something like that, where you really need a very fast, response that really takes place. You have to sense something, you have to compute, and then you have to respond all within seconds. So that's the kinds of things that we're interested in. And wearable devices too, that might be able to do all kinds of sense and response mechanisms.
So when you have this wearable device in the bottom left panel, it almost looks like a tattoo. Is it some kind of overlay, or is that like a cuff? I can't really see.
Yeah, so this would be some kind of a device, rather than, this would be a biohybrid device. So basically it would have, biology and electronics on a wearable system. So you can have cells that can sense, that interact with electronics, and maybe, tell you, what's going on, whether there's any kind of aberrant activity that's taking place in your blood vessels or so on. So that's one idea, is to do it on the outside, and another idea is actually to do it on the inside, so that we would be able to actually encapsulate cells, and it could be that we encapsulate cells with devices such as, maybe, I don't know, could be a pacemaker, or could be something that we implant subdermal. It could be something that we implant like in stents. So you can imagine all kinds of implants that we could do that are hybrid, meaning that bio and electronics, or ones that are purely biological. So, and some kind of an implant that is encapsulated by certain kinds of devices such as beads or something like that, where the cells would just be autonomous.
I was attending a talk with Doug Melton the other day. He was speaking about his, I'm sure you know him. He's over at Harvard and at HSCI, and he's he's developing his company Sema now sold to Vertex, and they're developing the implantable islets that they've engineered from stem cells.
Yeah.
And so, he was talking about how it's such a small size is needed, just the size of a pea to really incorporate all of the functional beta and alpha cells that are needed in the body. So you can imagine, that they're trying to figure out now where to implant this. And of course, how it has to be removable in case of teratoma.
Yeah.
But I just wondered what your thoughts are in terms of implanting. Could you even have a teratoma sensor, some kind of sensor in there that would be protective or alert to things going wrong in the system?
Yeah, so you can imagine building genetically engineered cells that can kind of survey the number of cells that are present to them that are engineered, so they can essentially do some kind of a count to find out, is there like an overgrowth, an undesirable overgrowth, and they might be able to report that to you.
Yeah.
So you can imagine, for example, you might be able to create, we're working now on embedding like a liver organoid within these beads, but also are interested in embedding a pancreatic organoid that would be maybe like around 5 millimeters or so, and be able to, in diameter, be able to embed that. And you can then imagine other kinds of sense and respond mechanisms that would continuously monitor for performance, function, number. So maybe if the pancreatic organoid that we engineered is not really performing properly, so we could have these controller cells that are detecting that there's not appropriate sense and respond mechanisms, and then report that to the physician, and let the physician know that there's a problem with the behavior, the performance, the architecture, the size, various kinds of things that you might be able to detect.
Yeah, and then of course it would be replaced, I guess, right? So that's really interesting. I wondered if you could comment a little bit about I was reading some of your work about the whole idea of this system working directly on immune cells. So for instance, right, the immune cells are sensing problems, and that then you get the miseducation of an immune cell, and then it attacks and creates the autoimmune environment. So just to speculate or sort of throw it out there, how might this system interface with immune cells that are either on the cusp of going rogue or have become autoimmune?
Yeah, it's a great question. So there's definitely been some significant progress in the field in terms of being able to manipulate immune cells, either to take them out of the body and being able to re-engineer them and then put them back into the body, or something that we're quite interested in is, can you engineer the cells in vivo, meaning can you keep them in your body and then reprogram them while they're still in your body, and you want to be able to make sure that when you engineer these cells, you can guarantee that you really are engineering the immune cells and not other cell types. So you need some mechanisms such as what we're calling classifiers to ensure that if you're going to genetically modify cells in your body, you're modifying the right cells. So the thing that we would ensure first is that the new genetic material goes inside the cells. It does a biological computation to detect, is this a right cell or not? And if it's the right immune cell, then it would make the modification. And if it's the wrong cell, the undesirable cell, that it would just basically leave it alone. So you need that initial level of safety to make sure that you're only modifying the right cell types. And then, then the question is, could you, detect that there's, like you said, there's a rogue immune cell that's maybe proliferating or attacking cells in undesirable fashion, resulting in some kind of an autoimmune response. And the other flip side to that is, can we also engineer, let's say we create a pancreatic organoid and embed that, can we also engineer our pancreatic organoids to be able to communicate properly with these immune cells and make, if a rogue immune cell is coming in, basically provide signals to that immune cell to actually stay away. And so this is, theoretically possible. It just has to be done very, very carefully, right? So for example, cancer is a manifestation where your immune cell is interested in destroying this undesirable growth, but these cancer cells are providing signals to the immune cells telling them to stay away. So things like checkpoint inhibitors or secreting some kind of immunosuppression cytokines or chemokines that tell the immune cells to go away. So there's a lot of dialogue.
Or not showing their MHCs.
Exactly. So when we do these modifications, it's absolutely critical that, as we're doing them, we're also maintaining safety, right? So if we're genetically engineering these, pancreatic organoid cells to not respond or to tell the rogue immune cells to stay away, we still have to make sure that we haven't also introduced, cancer. We didn't introduce teratoma, so that we need to build further safety mechanisms in there to guarantee that doesn't happen. So we didn't solve one problem and then create another problem. But that's kind of the beauty of these synthetic biology approaches, is that you can actually engineer the circuitry to create safety switches and so on.
Well, that's still one of the biggest issues, right? Is once you implant islets, you may have, great alpha cells, beta cells, everything's ready to go. You implant the islets, but still the immune system has a memory. And in those who have had type 1 for a while, that immune memory is going to be reactivated once it sees functioning beta cells. So yeah, there's a lot of work around sort of this invisibility cloak, if you want to call it that, and trying to get the implanted islets to fly under the immune radar so that the patient doesn't need to take the immunosuppressants. And it does seem that this switching, this sort of switching idea is just such a really interesting one. Like you said, I mean, you'd almost have to have the two switches. You have to put on the invisibility cloak and meanwhile be counting cells nearby. Something's got to be counting to make sure that teratomas aren't forming. So it's really a fascinating.
You essentially need to. Sorry, go ahead.
No, I was going to say it's super fascinating, and I also wonder about, one thing that's really interesting is that people that get, first display this one autoantibody GAD65. They typically have the DR3, DR2 HLA group. And so, the whole disease is getting more refined. It's more, understood about, who gets what and how the progression happens for different genetic profiles. So, I mean, you have to think about essentially it might be different combinations of sensors and networks that have to be addressed in different patients. But in terms of that HLA group, the DR3, DR2 getting susceptible to autoimmune, the first autoantibody arising is GAD65. What about a signaling intervention there? With that actual HLA.
With the receptor, yeah. But anytime you mess around with that HLA, then the question is, do the natural killer cells actually detect, the aberrant behavior? So that's one, so there's been really a tremendous amount of emphasis on creating hypoimmunogenic iPSCs, right?
Mm-hmm.
So ones that don't have, the HLA. And the problem is that, with biology, there's always this balance. There's always like trade-offs. There's always redundant pathways. And so what can happen in that situation is that natural killer cells, so rather, you won't get attacked by, let's say, CD8 T cells, but you will then get attacked by natural killer cells that are detecting a different kind of, aberration. And so that's the situation where you always have to think about what are the redundant mechanisms here, and can you then, so if you want to reduce HLA, can you still at the same time ensure that the natural killer cells are not coming in and recognizing you as well? So you need to find additional mechanisms there.
Yeah.
So it's like, you kind of have to find that the homeostasis and the trade-offs between all these redundant and pathways that operate to make sure that they've been developed in your body to make sure that one thing goes wrong, then there's a backup mechanism to overcome that. And that's where we need kind of the multi-input approaches and multimodal approaches.
Multimodal. And it does seem that this whole idea of the implanted, islet or beta and alpha cells, along with some of the systems you're talking about, might be the best way forward.
Yeah.
I just wanted to open it up to anyone who's interested in asking a question. Look in the chat. You can drop something in the chat if you're interested. Does anyone have a question at all? I guess we could say how long till some of these systems get to market?
That's I would like to say the $1 million question, but it's really a $1 billion question or more. Yeah, I mean, nobody really knows. I think that under kind of our most optimistic timelines, I mean, could be a few years, where maybe not fully to market, but certainly making their way through some clinical trials. So depending on the level of complexity that you're talking about, there's some things that we're interested in. So in terms of organoids, that optimistically maybe in 2 to 3 years we could begin to think about the entry towards phase 1 clinical trials. So it really depends, and I think that there are different companies at slightly different stages. Some of them I think have IND enabling studies. Some of them are really thinking about phase 1 clinical trials, and then when it's has to go through this process of the vaccines have been unbelievable in terms of like the speed at which we've been able to take the messenger RNA, put it through design phase 1, phase 2, phase 3, and get it out there in a year. That's the timelines that we're talking about here for these kinds of cell-based therapies are certainly much longer. So whether they're going to be 7 years under kind of optimal conditions to the point where they pass the phase 3 clinical trials.
Yeah.
But,
and what other laboratories? No, and it's an exciting times. What other laboratories are doing this work, interested in this space, and your collaborators, just to sort of throw out some names and locations, just in case anyone who listens to this now or later has interest in reaching out for collaboration or just connection.
Yeah, I mean, I'll do some free advertising, if you will, to the Mammalian Synthetic Biology Workshop that has a lot of kind of like-minded people that are excited about the prospects for synthetic biology in particular. I'm sure, over the different sessions you guys have had here, that you've been able to discuss people, they're working with islet cells and, beta cells and so on, working on the kind of the biology aspects of those I would say with synthetic biology, the community is really active and growing nicely. We just had a recent mammalian synthetic biology workshop, and that might be still archived for people who are interested in looking that up. And we had a specific session on the creation of genetic circuits for stem cell differentiation and for organoids. And then we hope to have the next version of the mammalian synthetic biology workshop in Edinburgh in Scotland. That would be next summer, I think July 28, 29,
Yeah.
if people are interested.
Yeah, I see that right now, and I'm going to retweet it for you guys. And yeah, that's fascinating. How big is that group and that consortia?
Yeah, we've had about 300 people participate in these workshops.
Wow.
So it's been it's been just a really fun community, and I would, if people are interested about learning how to create these genetic programs, the sense and respond kind of mechanisms, and what that might do, how that might help, I really would recommend finding out more, looking at some whether there are going to be some online presentations that were recorded from these workshops, and then also possibly coming to visit in Edinburgh to find out what's going on.
Yeah, it looks like it's going to be live, so that's great, fantastic. Well, we'll definitely share that with our community, and thank you again for this fascinating talk. The work you're doing is really, it really has a footprint in type 1 diabetes. Hardcore science, and hopefully, with your work intersecting with those of the work that's happening with islet implantation, robust systems can be developed and things can move forward. So thank you again, Ron. Really appreciate talking to you, and hope you have a great rest of your day.